So, Alex, I'm really very flattered and honored that you asked me to introduce you. Yeah, I have known Alex for over 40 years. I first met Alex at a Gordon conference. I think it was 1965. And I used to go to these nucleic acids Gordon conferences, very popular. And Alex was already an established fixture at these conferences. The conferences were heavily subscribed, but attendance limited to about 150, but many wanted to go and many could not. But Alex was among those who did not have to apply to get in. So there was always 5 or 10 spots left at the very end for late applicants, and late applicants being people like Alex and Ochoa and Lipman and Crick and others. Yeah, so I got to know Alex better when I moved to MIT, and Alex had already been here for at least 10 years before that. I think you came here in 1958. Yeah, so it was 11 years before, and got to know Alex very well since then. Year after I arrived here, I was asked to teach a course with Alex. It was a pleasure. We taught that course for many, many years, and that course has now morphed into the course 777, course on nucleic acids and proteins, that Alex still comes back every year to give a lecture for us, a three-hour lecture with almost no break, although I think we persuaded him to give students a 10-minute break. And I think it's a real treat for our students. Scientifically speaking, Alex's life has been really one major achievement after another. It's been a lifetime of achievements, beginning with the structure of collagen and polyglycine to discovery of RNA hybridization, RNA DNA hybridization, to triple-stranded nucleic acids, to the discovery of polyribosomes, to the crystal structure for the first time of double helical nucleic acids, to the crystal structure of an RNA molecule, that of transfer RNA, which remained the only RNA molecule of known structure for many, many years. And then that led to the discovery of the left-handed DNA, to which Alex coined the term zDNA, and then on to zDNA binding proteins, and then the role of zDNA binding proteins in messenger RNA editing to generate neurotransmitter receptor subtypes in the brain. So the list goes on, and as you can see, there's no sign of slowing down, right? So that's really amazing. I think that what I find most remarkable about Alex is that he makes it all look as if it's completely effortless. You don't have to do a thing. Just things just happen to him, and that's really amazing. And you have to be among the calmest of the people I know, Alex. So that's So another really remarkable thing about Alex, looking back, is his really uncanny ability to foresee and predict things that are going to happen. And I think part of the reason for this is Alex's tendency to think out loud. But unlike most of us who would think out loud and imagine things but never put it into print, Alex puts them into print. And so I came across publications from Alex in 1961, 62, that suggested that RNA comes before DNA, must have come before DNA. And I think most of us believe that's what happened. And that also suggested, speculated, I should say, that RNA could act as an inhibitor of another RNA molecule. And that was through complementary base pairing. And that echoes of microRNAs, siRNAs, and antisense RNAs that we talk about these days. So that's really, really remarkable. I thought about my experiences with Alex over these years, I thought there could be a couple of episodes that might be of interest to this audience. And let me just tell you about these things. Again, a year or two after I arrived here, Alex said, we should write a grant together. And so we decided to meet in his office at 7 o'clock one night. Alex, as you know, works late. 7 o'clock, and we talked for about an hour, maybe a bit more than an hour, about what the grant could be about, and what sort of things to emphasize and all that. And all of a sudden, Alex stands up, and he picks up his dictaphone, goes to his sofa, lays back on his sofa, and starts to dictate. The whole grant, well, more or less the whole grant. And the next day, there was a reasonably good outline of a grant already typed. And I think it's the fastest grant writing that I've witnessed. Another experience was writing a review with Alex for annual review of biochemistry. This happened the year that I was going on a sabbatical. So Alex and I met 2-3 times just to discuss the topics to cover and things like that. And then I left, and the rest of the decisions were made on the phone. 3 or 4 very, very long phone calls between the 2 Cambridges. And the review was essentially written, and I sent my review written longhand to Alex, and when I came back, it had been submitted. The review was published in 1976, and I have to say that it's still quoted, not just by us, Alex, but so. And I want to end on a personal note, Alex, that you have to be among the most kind and generous of the people I know. I have been the beneficiary of many, many invitations to parties and dinners at your house. These are really very relaxed events, relaxed affairs with lots of people from all walks of life coming and going during the 3, 4, 5 hours, whenever. And it's a wonderful opportunity to also meet lots of guys from Harvard, because if you want to meet people at Harvard, go to Alex's parties and you'll meet them. So it's really full of hospitality and just very enjoyable. So Alex, I want to thank you, and I hope I'm not taking too much of your time.
No.
And so we're ready to hear from you.
Well, the group here is somewhat bimodal. A number of you are in the same age group as I, and many of the things I'll say about olden days will not be so remarkable. But a number of you started research, were not born, I should say, when I started doing research, and so some of this will seem very ancient. This entire discipline did not exist around 1950, when I finished medical school in '49, and then went as a postdoctoral fellow with Linus Pauling. My interest had always been in doing research. I didn't quite know in what direction, but I do remember while reading a pathology text and in medical school, and I must say I found all of medicine very interesting. It's really the detailed biology of one organism. On the other hand, I remember sitting there and saying, I don't think I can bear to go through life without understanding relativity theory. Well, I'm looking forward to learning relativity theory. But in around 1950, to a first approximation, nothing was known of the biology that you're familiar with. A few years before, Avery had, of course, written his important paper identifying DNA as the transforming factor. Hardly anybody knew of that paper. It made very little impact. I went to Pauling because he had just made these fantastic discoveries of a molecular disease. I mean, there was a riveting paper in Science saying that sickle cell anemia is a molecular disease. And this shocked everybody. Today, you'd say, well, of course it is. What else could it be? But in those days, the idea that a molecule, that you could identify a disease in terms of something that happened to a molecule, was absolutely astonishing. And it was very much a paradigm change. I mean, you can talk about the view of disease before and the view of disease after. Well, I arrived at there just when a couple of years after this discovery was made, and Linus, well, he was a remarkable man. He, first of all, I walked in to see him dressed, you know, coat, tie, very much a sort of uptight East Coast, you know, medical person, and walked in, and there was this fellow with a big smile and the most flamboyant Hawaiian shirt I'd ever seen. And so I could see this is quite a different world. He was very welcoming, and he said, you know, he said, we talked about what to do, and he said, I'm sure there are other diseases like sickle cell where the hemoglobin is abnormal and causes the disease. Why don't you have a look? So, using my medical credentials, I went to the Children's Hospital in Los Angeles and began examining different samples of blood from children with different hematological diseases. I wasn't very successful. I did discover a young 13-year-old boy who lived on 100% fetal hemoglobin, which was quite remarkable at the time. And but most of that work, but didn't go anywhere. I started working on theoretical chemistry, calculating valence state energy levels for carbon. Now this is before we had computers. We had adding machines and not much else. Lots of laborious calculations and mostly approximations. And I remember one evening Linus had a cold and didn't come in. And I talked with him. He said, well, come up to my house this evening and we'll talk. So he lived up on the hill in Pasadena, and I went up. And we talked, and he showed me a large volume that just came from the Faraday Society, a symposium on chemical theoretical calculations and about approximations that could be used. You couldn't solve the Schrödinger equation, but you made various approximations. And he took that book, looked at it, threw it down, he said, it's like whipping a dead horse. And he said, you know, he said, in the 30s I thought I could solve this problem in a closed way, that is, solve the equations. But he said, I couldn't. I made approximations, and now people are making approximations on the approximations. And let me point out, my calculations were based on approximations. So he concluded that the problem could not be solved in a useful way. When I left him, I thought to myself, well, there's Linus Pauling, and he can't solve the problem. What am I doing working in this field? Because I know that I don't have his talent in this area, and so I switched over. I began studying structure, because one of the things he told me early on, almost axiomatic. Look, he said, if you want to understand function, biology, structure is 90%. If you know the structure, you can get the other 10%. But if you don't know the structure, it's very difficult. And I took that very much to heart and decided I would learn how to do structure. So Linus at that point had already solved. He took over as editor of an obscure journal nobody had ever heard of, called the Proceedings of the National Academy of Sciences. Nobody read it, didn't have anything in it. And he brought out one issue of 8 of his papers, which described the alpha helix, the beta sheet, the gamma helix, structure of collagen. I mean, a breathtaking array. And half of it was correct, which was astonishing. And how had he done that? He did that through a method called the Stochastic Method. I think that was the name. Anyhow, what it meant is the following. You take a structure, first of all, which has regular elements. So that would apply to any polymeric biological compounds, polyamino acids, polynucleotides, or polysaccharides. You fix on some experimental observation, and you build on that and build a structure that incorporates that. And if you have the right intuition, and I would say luck, you get the right answer. So he, very early on, he had worked on the structure of polypeptides. And at that time, actually, the geometry of the individual molecule was not known, the individual amino acids. And nothing, well, very little was known about peptides. But he started a program of doing the structure of proteins and amino acids, crystallizing, solving. He had a large group doing that. He was intrigued by the regular patterns that you had if you took a hair and shot an X-ray beam, out came a regular pattern, telling you that there was some kind of a regular fold underlying it. And he made his best guess about the geometry of the peptide chain and the peptide bond, built a structure, a helix. It didn't quite predict what was in that pattern. And so he put it aside. And one reflection, off meridional, didn't quite agree with what he predicted it would be from his model. So he set it aside. Spent a couple of years looking at the peptide bond. And that was a crucial point. Because he knew theoretical chemistry, he understood when the first one was done that the peptide bond was shorter than it should have been if it were a single bond. Therefore, it had double bond characters, which meant that the carbonyl and the amino group would be planar. Now, about the same time, another competitive group, and let me just say that Pauling had a long history of competition with Willy Bragg in England. He had visited him once in the 30s, and they were both interested in the structure of minerals, and Bragg and his group were laboriously working out the structure of minerals, one by one. And each one, in those days, it was very difficult to solve these structures. No computers, and but Linus came in, looked at what was going on, thought about it a little bit, then went back and wrote a paper on the general structure of minerals. Minerals are made of ions. They're not, and the question is, how do you pack ions together in a lattice where the overwhelming interaction is electrostatic? And then you have the question of how big the ion is. So he wrote this paper about the general structure, a very subtle paper, but if you followed it, you could figure out the structure of all the minerals, just like that. And people were skeptical, and Bragg worked out a few more, and indeed, Pauling was right. So this is a backdrop to the fact that while he was working on these peptide structures, Bragg and his colleagues were working also on peptide structures. So a paper came out in the Proceedings of the Royal Society. Kendrew, Bragg, Kendrew, and Perutz had cataloged 83 different coiling helices of peptide chains. Pauling wrote a paper about one coiling, the alpha helix. What had happened, not being chemists, Bragg, Kendrew, and Perutz had gone to the professor of physical chemistry and asked him, well, will this peptide bond be planar or not? His advice, it's a marvelous case, his advice is, it'll have any angle you want, but not planar. So all of the things were wrong. Anyhow, it's a very good lesson in there. Don't take people's advice. Anyhow, all of this set the background for what later became another step in this competition with the structure of DNA. But anyhow, I did some work with some single crystal work. We solved the structure of ferrocene, kind of sandwich molecule with an iron in between. And I did that with Jack Dunitz and Leslie Orgel, who were there as postdocs. Couldn't have done it without my wife, because in those days, you measured intensities by looking at a spot on a film, and then you had a little strip, and on that strip you calibrated different exposures. So you had an increasing exposure. And what you did is you took that spot and you moved the strip back and forth until it matched, and then you assigned a value to the intensity of that spot. And my wife turned out to be very skilled at doing that. I had much less skill and not enough patience. But anyhow, the structure came out. And it came out in, it was a three-dimensional structure, which is, nowadays everything's three-dimensional. But in those days, most structures were done in projection. In one, you look down and you see it in 2 dimensions. But this was done in 3 dimensions, which was important. And I worked a little bit with Dick Marsh on the structure of silk fibroins. That is the silk structure, which is quite interesting. Linus at that time became interested in DNA. Why? Well, it was there in the nucleus. But let me say as a background, Mursky, a man from Rockefeller, a very good physical chemist, had come to take a sabbatical with Linus in the mid-30s. They wrote a very good paper on the importance of hydrogen bonds in protein structure. Mursky was a specialist in nucleic acids. He told Linus with great confidence that the basic proteins in the nucleus, those are the genes. There are nucleic acids there. They probably are a scaffold for holding them. So even though the Avery paper was there clearly indicating that the DNA was the genetic material, it was not widely known. And Mursky in particular, who was a colleague of us, of Avery at Rockefeller, completely denounced the paper as being incorrect, and he wasn't a very nice guy. He felt as if, I suspect, Avery was poaching on his territory with the nucleic acids. But so I don't think Linus looked on the nucleic acids quite the way Jim Watson and Francis Crick were looking at it, because Jim had been taken his PhD with Salvador Luria. They knew about the Avery paper and understood it in great detail, and were convinced that DNA contained genetic information. And so he was working, they were working there. But anyhow, at one point, Linus came in, and Jerry Vinograd was a very good physical chemist who had been there too. He came into a room where Jerry and I were sitting and talked about the structures he had finally deduced for DNA. He was looking for, by his method, some physical clue. And he found it, he thought, reading a paper about the structure of some mineral in which phosphate groups were organized in a helical array. And so he said, well, if that's true, could he build a sugar linkage from one phosphate to the other with a base sticking out? And the answer is he could. And so he ended up with this DNA which was triple-stranded and with the bases on the outside. And he had done it based on calculations of density, which weren't very sound. And I remember his describing it to us. I looked at it. I didn't think very much about it, but I was a little concerned. What are those bases doing sitting out there? You know, what's holding them out there? They're just sort of flapping out there. And anyhow, he published it, and immediately it became apparent he had made a big mistake. The thing is in water. These phosphates have to be ionized. They would never hold together. He was looking at a structure of an anhydrous phosphate, and it was completely, he had misfired. Now the odd part of it is, Crick and Watson were working a little bit on DNA, illegally in the sense that the MRC has, the Medical Research Council, had an agreement. They set up a unit in Cambridge, England, for the structure of proteins. They set up a unit at King's College, London, under J.T. Randall, on the structure of fibrous materials, which included collagen and nucleic acid, DNA. So they were kind of poaching. And after they did this triple helix, they looked at it, and it was clearly wrong. And the people at King's College learned that they had done this. They complained to Bragg, and Bragg forbade them to work on DNA. So they were, you know, chastised, and were the dictate came down, you can't do it. Now in those days, now it's an interest, you know, things are different today. You can't imagine. You can't imagine a professor saying, you cannot do that. You know, that sounds so odd. But it was quite the norm in those days. And in fact, even odder in today's world is people would write a paper, make a discovery, and then in the paper claim that this field is theirs, which means don't poach. And that of course doesn't happen today. But but then, a few weeks later, out came Pauling's paper, which he published. And Bragg saw that, and the old competitive juices began flowing, because they had really been sort of, he felt badly about his 73 structures that he had published for the alpha helix and so on. But he felt more annoyed that Linus would solve DNA than that Randall would be unhappy. So he gave Jim and Francis the okay. Go ahead, you can go back and do DNA. Well, 3 weeks later it was done. So you have to ask, you know, how big a problem was it? They of course had Rosalind's diffraction pattern. Francis had written a paper with Vand and Cochrane, Crick, and Vand, on the diffraction of objects in a helix. And in it, they had understood how you could interpret a pattern and understand what a helix is built like. And here was this pattern, and it was clear what was going on. And anyhow, the structure came out very quickly. Linus bumped into me in the hall. He'd received a letter from his son Peter. Peter said, Peter wrote to him saying, two English guys, Jim is hardly English, but have proposed a model with the bases on the inside, 2 strands. And Linus had told me that. I thought that was interesting. I remember I went home to bed and I couldn't sleep. So I came in, I got up and left and came into the lab around 1 or 2 in the morning and went to the room. There was a room with models where we could build. These were brass skeletal models. And we had some cutouts for the nucleic acids. And I should say, we had the correct tautomeric forms because Linus had worked that out. And as you know, Jim had been working with models trying to pair them, but he couldn't solve them. And Jerry Donahue, who was a postdoctoral fellow at Cambridge, who had just come from Linus's lab, had knew all about the tautomeric forms and said, "You've got the wrong tautomer here. It's a keto form, not an enol. And if you'd done the enol, you'd never get pairing. But with the keto, it locked together." So I went into the lab and I said, well, how could you get a regular pattern? I looked, and in a few minutes I could see, yes, you pair these 2 together, and then I could build it, and there it was. And then the paper came out about 3 weeks later, and I could see that was it. So, but then I had become interested in RNA. Well, the first question I asked was, the big problem is here are these 2 strands, they're all coiled together. How could they come apart? And I said, well, maybe it's possible that they go right-handed and then they turn left-handed for a while. So there I was working in that model room, trying to discover what was going on. Could you build a left-handed DNA? And at one point, Linus stuck his head in the door and said, "Alex, I want you to work hard on that problem because I like all important discoveries to come out of Pasadena." Well, let me say that had been true, barring the DNA business, it had been true for quite a while. So he wasn't saying something that sounded so unusual. Anyhow, I convinced myself that you could not build a left-handed double helix, i.e. in the bDNA format. So, and in fact, if I jump ahead 30 years, I was at a meeting in early, about 1980 or 82 in Italy and Linus was there. And my paper was about left-handed DNA, zDNA. And I reminded him of this issue where he had told me, I said, I'm sorry I didn't succeed at that time. It took me a longer time to solve the problem. But he, anyhow, only a small number of people were involved in this kind of biology. At that point, I became interested in, what about RNA? You know, if you look at the Watson-Crick paper, Francis put in there quite correctly, you cannot form this structure with a ribose backbone because the van der Waals crowding due to that 2' hydroxyl would prevent it, and that's completely true. And so I started working on that. In September of '53, Linus had a conference on the structure of proteins in Pasadena and nucleic acids. All the people from Britain came over. I mean, Bragg, Kendrew, Perutz, Watson. Well, no, Jim had come back earlier to resume work with Max Delbruck. Francis, Bill Asprey, Ian MacArthur, a very long string of people from Britain, and a comparable group. So there is this wonderful photograph for those who like history of all these people lined up, and it's a kind of early history of proteins and nucleic acids. At that point, what I was doing is taking fibers. Well, let me back up a minute. In December of '52, Linus, having realized he's made a mistake with the triple strand, writes a memo to me saying, well, first he wrote a letter to Morris Wilkins asking, there had been a conference of the Royal Society on Protein Structure to which he was invited. He was having trouble with the State Department because he had this outlandish idea that testing nuclear bombs in the atmosphere was bad for your health and we should stop. And they considered he was a communist for doing this. They wouldn't give him a passport, so he couldn't go to the meeting. But at the meeting, Morris Wilkins presented a number of pictures of diffraction patterns, including one that Rosalind had made. Rosalind by then had left King's and had gone to Birkbeck, so she was no longer working in that area. But he wrote asking if he could see the pictures because he said he was unable to get to the meeting. And Morris wrote back saying, declining to send them, saying he hadn't finished interpreting them. Makes you wonder, since he had presented them in public at the meeting. But he was afraid, quite justifiably, that if Linus saw this picture, the structure would come out. So in this memo to me, he asked me, would I take some DNA and make fibers and get a diffraction pattern? So I started doing that. And I have some of these early pictures. They're not bad. They're not they're not in Rosalind's league, but good enough so you could see the patterns beginning to come out, the helix. When the paper came out, I realized I stopped that, and then I began pondering, can RNA do it? So I began taking RNA fibers. And at this meeting, Jim became very interested, and he had come back to work with Max Delbruck, but he was less interested in phage and more interested in the question of RNA, because we knew that RNA had to do with protein synthesis. So we said, let's work together. So we collected samples from lots of people. We wrote them letters saying, would you send us a sample? We're trying to study this problem. We probably got 40 or 50 samples. They all looked the same, and the resolution was such that I couldn't tell whether it was a helix or not, a double helix. We published a couple of papers, but it was inconclusive. But it did have one odd thing. No matter what the base ratios were, the pattern was always the same. So that was sort of a puzzle, because in DNA, the base ratios were always 1:1, and they all gave the same pattern, but in RNA, that was different. So at this meeting, I met Francis, and he was very interested in the subject. He said, well, in Cambridge, we've developed a new, very powerful X-ray source, a rotating anode. And he said, if you begin to get good results, come over and use this machine because it'll give you a good, quick pattern in a short time period. And I then left NIH in '54. I had been given the job of setting up a section at NIH. Building 10 was just built. And I went there. I should say, I had joined the US Public Health Service because I was liable for the draft. Since I'd gotten my I had been in the Navy for 3 years during the war, but that did not insulate me from the draft, because the Congress passed a law saying if you got an MD degree and you hadn't served in the Army or in the military as an MD, you were eligible for the draft. And I considered that was immoral. So I joined the Public Health Service. We were called the Yellow Berets. Well, there are lots of people who went there then. But anyhow, I went there to set up a lab. I discovered I had to hire some people, and a group of us collected and started working. And about that time, Severo Ochoa discovered with Marianne Grunberg-Manago polynucleotide phosphorylase. You feed it nucleoside diphosphates and you get an RNA chain. And this was ideal. They were long enough so they could be well oriented, and I could get a diffraction pattern. So I wrote to Francis, and we went over to England for a short visit. But while we were there, he put me up in his house, which was very generous of him, for a short visit. The short visit extended to over a half year, because one morning, and it was a Saturday morning, and I came down to breakfast. Odile had slept late, his wife. She didn't want him to come down, so she sent their young daughter with Nature that had just arrived in the mail on Saturday up to give it to Francis in bed, knowing he'd stay in bed long enough for her to prepare breakfast. So later he came down, and I said, anything interesting in Nature? He said, no. Well, he said, there might have been there. There's a new form of polyglycine that the people at Courtaulds had seen. They made it. They didn't know what it was. It had a different infrared spectrum. I should say normal polyglycine is one where the glycine chains are fully extended. And if you look at them, they're in a helix, twofold helix. That is, to get to the next residue, you rotate 180 degrees, and then like that. So this new form was different, and they had a powder diagram, lines. And as we sat there, we said, you know, we should be able to solve that. Solve meaning make a model. So we went in, and we went in at 10, and by noontime, we knew we had solved the problem. How had we done it? Nothing very profound. We said, well, this is a two-fold screw axis. Let's look at a three-fold screw axis. So we put 3 of them in a turn, and then we saw the glycines would all hydrogen bond together. So you made this big lattice in 3 dimensions, nicely hydrogen bonded. And we could predict not only the reflections from this, but their intensity because of how many atoms were in the plane. So we knew we had the structure. So then Francis had the idea, let's publish it in next week's Nature, write it up and get Bragg to send it in. And that seemed like such a nice idea. And then he thought about it again. He said, on the other hand, the people at Courtaulds, they'd be very unhappy. So we decided, no, what we'll do is invite them to come and look at the structure. And they did, and they nodded, and they said, yes, yes, that's correct. And then we sent it in. And but as I looked at that structure, it was quite interesting because I noticed that the NH bond was coplanar with the CH bond that would normally be where you'd put another methyl group if it were alanine and so on. And because those 2 bonds were coplanar, I realized you could build a pyrrolidine ring around there. And because of my exposure to Linus and his work on collagen, he had proposed the structure for collagen, but it was wrong. And collagen, if you took a rat tail, a tendon, and stretched it out, and took an X-ray picture, you got a marvelously detailed picture, quite unlike anything else. And so I remember Francis and I went out to have tea one afternoon, and I described this to him. I said, you know, it may be that we could take 3 chains out of that lattice and build a collagen molecule. He had been thinking along the same lines, so we went ahead and did it. In that structure, the distance between residues was 3.1 angstroms. But we knew from collagen there was a meridional reflection at 2.86. So we simply took those 3 chains and twisted them somewhat. And when you twist them, the distance of the residues along the axis goes down. We twisted them to 2.86. And it looked perfectly well. We knew that collagen had sequences like proline, glycine, proline, and then hydroxyproline. So we built that model, GlyPro-Pro, took a great diffraction, took a projection. We had a light, very sharp arc at one end of a long hall, and put a piece of paper on the wall, little circles for each atom. And then, with a reducing pantograph, took a little piece of aluminum and drilled holes, each hole representing 1 atom. The reducing pantograph, you'd move it to the next hole, and this little arm would move a small distance. So out with this pattern. And then we went, Arthur Elliott at Courtaulds had a very good optical diffraction instrument. We went, put that in, took the diffraction pattern, and there was collagen. You know, just very exciting. Anyhow, that's why I stayed another 6 months. However, during that 6 months, we talked a lot about RNA structure. And we talked a lot about these polymers. But nobody, including myself, said, look, you've got polyriboadenilic acid and polyribouridylic acid. Why don't you mix them together? Nobody thought of doing that. It seemed like such a far-out thing. And it wasn't until 6 months later, when I was back in Bethesda, that for reasons that I don't know, I mixed them together. And immediately I could feel that the solution became very viscous. And when I pulled them out came this fiber. We took an X-ray pattern, and my God, there's a double helix. Not the DNA double helix, a different one. And this was the first hybridization reaction. Can you imagine that two very long polymers combine together laterally with specific hydrogen bonding to self-assemble into a double helix? That was such an astonishing thing. A number of people told me, we wrote a short note about this, a number of people told me this was quite incorrect. First of all, I remember when I described this to Herman Kalkar, I said, Herman, walking along a corridor at NIH, Herman, poly A, we mixed poly A and poly U, and we got a double helix. And he said, what? He said, without an enzyme? It seemed to him impossible. And other people said, these polymers are thousands of angstroms long, and they're all random. They'll never untangle. And furthermore, they're all highly negatively charged. They'll never come together to make a double helix. And that, so there was a great deal of skepticism. But nonetheless, it was quite clear that it was correct, and that was the beginning of hybridization. I see that I've now used my hour.
You want to take some questions, Alex?
Hmm?
You want to take some questions?
Oh, I'd be glad to have questions, yes. I didn't want to make this a monologue. But I'm only, look, let me just say that if you want to hear the rest of the story, I did write a review at Annual Reviews of Biochemistry about, it's a scientific autobiography of what work I've done, so you can read about it in there if you want. But I concentrated on the beginning. Beginnings are always odd because you never know what you're going to do or what you can do. And I must say, when I left Caltech to go to NIH, I felt very insecure. I concluded that I'd done nothing of any value at NIH, at Caltech. And so I was, you know, it wasn't clear to me that that doing research and making discoveries was my thing. Although I do remember we had a meeting on the structure of collagen at about a year or so later in Britain. And Linus was invited to give an opening speech. And he gave a very nice talk. And at the meeting, having tea at an interim, a group of English people were in a circle, and I was there. And one of the Englishmen asked Linus, is it true that Dr. Rich worked with you as a student? He said, yes. He said, Alex spent 5 years in the lab. He didn't accomplish very much, but he must have learned a lot. I took that as a great compliment. The and just parenthetically, Francis and I published the structure of collagen, which of course was another King's College. That was their their thing. So this just exacerbated the problems. But yeah, no, let me ask for questions about this early era.
How hard was it to switch? You were in a lab, in Linus's lab, doing calculations, and then all of a sudden you said, no, I'll switch to, it's fairly large change, even within a lab. Was there any resistance to your doing that, or was it pretty much, he would say, do anything you want to do, Alex?
Oh no, Linus, he was, his attitude was, he can make suggestions, but you do what you want. He was very good that way. And I must say, a lot of people did very good work, and some people did poor work. And I've actually always adopted that attitude too. In other words, I can't imagine telling a postdoc you must do that. My attitude was, postdoc would say, well, I'd like to do this. And I would encourage them, or maybe, you know, amend it slightly. But I think the spirit in the lab was very, very good. What it meant, if you came in the lab and you did well and you left, you knew you were able to do research because you did it alone. You didn't, there was very little tutoring. But of course, some people came and did nothing, and then for them, who knows? In my case, when I left, it wasn't clear that I could do anything, but.
Why did you decide to come to MIT?
Oh, well, I'd been at, mind you, this was now in the early 50s, middle 50s. At that point, this is now, tomorrow, I remember where I was 50 years ago tomorrow. I was in Bethesda on a big baseball field in the middle of the night, looking at the sky, and we finally saw the Sputnik coming over. And that was such a transforming event, because what happened then is suddenly the nation decided we better be serious, and they decided to fund research and to build up universities, and you know, the whole training boomed ahead. So within a year, now NIH was the place people would come for looking for faculty. So within 1 year, I got, I think, 8 offers of, and it wasn't that I was so great, it's just that NIH was a good hunting ground. And they were East Coast and West Coast, chemistry and biology, and full professor, associate professor, I mean, different levels. I decided I would only make a decision based on what I thought were the prospects for growth. I really wanted to get involved with students. That was the main incentive for coming. Because although I'd built up a very good group there, David Davies, Gary Felsenfeld, Don Bradley, Gunther Eichhorn, very good people. I felt it would be more interesting to start working with younger people. And so when I came to MIT, there were 8 people in biology. Most people at Harvard didn't know MIT had a biology department. When I told them I was going there, they were very surprised. But I had talked to Vicky Weisskopf and to Jerry Wiesner, and they felt they wanted to make a push in this new science. And I learned that in Vicky's case, he had been prodded by Max Delbruck, who said you ought to get into that area. And so it looked to me like the prospects for growth here was considerable. And there was only one person in the department, a friend of mine, Cy Leventhal, had come a year earlier, and he told me, let's go. I mean, come on, it's a good place. And fortunately, Erwin Sizer was first temporary chairman and then chairman. Erwin was a biochemist, very much committed to having people who worked at the molecular level. So in a sense, everyone there could then become what later was called a molecular biologist. A few people in the department were not, and they rapidly left. But it was just very exciting. And then Salvador Luria came for a sabbatical, and Cy Leventhal and I worked very hard on him to stay, and he decided to stay. I remember we spent a lot of time in the swimming pool with him because he liked the swimming pool and he didn't have one in Indiana. So anyhow, he came, and then well, shortly after that, lots of people, Boris came, and the thing took off. But my reasons for coming was it looked like a place where considerable growth could occur, and I knew the quality of the science here was very high, and that was the focus. So I every time I thought about going on a sabbatical, I would say, well, can I do science better there than here? And the answer usually was no, so I stayed here. Never went on a sabbatical. Well, these were all the beginnings, and the actual discoveries. Well, I can read you one thing, okay? So in 1956, we discovered poly A, ribo A, and ribo U made a double helix spontaneously. But everyone at that time was saying, well, you know, DNA is supposed to make RNA, and RNA makes protein. How can that work? How can you transfer information from DNA to RNA? So I said, well, you must do that probably by making a hybrid helix. So I learned, I'd met Goban Khurana when I was at NIH. He'd come to visit Arthur Kornberg. And when I saw that Goban was able to chemically make oligodeoxy thymidylate, that is, polymers of short lengths of poly-DT, I wrote to him, and he sent me a sample. I told him what I was trying to do. I then fractionated the sample and took the largest, the longest fraction, combined them with poly A, and out came, I was looking at the hyperchromism, and I could see you made a 1-to-1 helix, this was the first DNA-RNA hybridization. I published that paper. It was one of the first papers I published when I came here. and that was 1960. And at that time I had, I invited Antioch students to come. They have a work-study program where they would go off for 3 months and work somewhere. So I offered them jobs. So I had the Antioch student, he made all the measurements, and I thanked him at the end of the manuscript. That was Mario Capecchi, who arrived as a chemist and left as a biologist. And then later on, Joan Stites came, not knowing what to do, but I put her to work making RNA, and that turned her on to molecular biology. But shortly after that paper, I was invited to give a paper to a growth symposium, the Society for the Study of Development and Growth, a book they published, Molecular and Cellular Synthesis. So in it I described the transfer of information between nucleic acids. And this was written in 1960 and published in 61. So it was written before mRNA was discovered, and more particularly before the nomenclature. But the idea was you had to work this way. But DNA made RNA, and the hybridization told me how that happened, by making a hybrid. But then there was some evidence that you had a DNA-dependent RNA polymerase, but it had not been purified enough to know what was going on. And in this I discuss about how you can, here's this enzyme, it's probably copying DNA to make RNA. But then I say, is it copying one strand or two? So I say here, let us assume for the moment that 2 strands of RNA are made from DNA. These 2 RNA strands are, of course, complementary to each other. What is the physiological role of these 2 complementary RNA strands? It does not seem likely that both of them go on to manufacture a protein molecule, even though it's not impossible. But a mechanism such as this seems to offer no advantage. There is, however, an interesting possibility in that this may be part of the control apparatus for turning on or off the synthesis of a given class of proteins. One might imagine that one RNA strand is continually used in protein synthesis, while the other strand is used to control the rate at which the operating strand, see, the word messenger hadn't been invented then, at which the operating strand is made available. The synthesis of a given protein would be impeded by the formation of a specific 2-stranded RNA molecule, which then inactivates the template for protein synthesis. I leave indeterminate the method for inactivation. Alternatively, hydrolysis of the complementary or control strand would then release the operational strand and enable it to participate in the synthesis of proteins. In other words, what I'm saying there is the information content of the complementary strand might be used for regulating protein synthesis. And now fast forward 40 years, 43 years to microRNAs or RNAi, and they're back to this idea of using the complementary strand to modify whether you make it or not. But this was an idea that seemed quite reasonable to me at the time. I'm sorry to say I forgot it in the intervening years. And only recently I was asked to write a reflections article for the JBC that was celebrating its 100th anniversary. So I decided to write on the background of the first DNA-RNA hybridization. And then I happened to look at the next folder where this paper was, and I read the paper, and I almost fell off the chair. But it shows you some ideas. If the some discoveries can be made prematurely, and then they die.
So, Alex, journals these days discourage speculation in any form. How did you get away with speculating so much?
Well, good news and bad news. The good news is I could write what I wanted. It was an invited paper. The bad news was it got put into a book, not into a journal. Never indexed, and then it effectively disappeared. And that's also true. I wrote an article a year later on expanding information transfer, in which I described, and I talked about the origin of life, and I described it saying, well, it seems quite likely that life began with an RNA molecule and a primitive method of replicating an RNA. And why the focus on RNA? Because RNA clearly is able to replicate and contain genetic information, but it also directs the synthesis of protein. So this seemed to me quite reasonable. So when 9 years later, an article was published that RNA was the origin of life, it seemed to me old hat. But again, it was in a book, so it never got cited. So, no, I think there's nothing wrong with speculation. I put speculation in all the time. Nice to have a little bit of data behind it, but not entirely necessary. Well, I'm afraid I've talked for longer than I should, so so far, it seems to me, so far I've got through the first 5 years of my research program.
Thank you.
Okay.